Well, I'm very happy to be here. Thank you, Pavel, for the invitation. I want to share with you, before starting the presentation, that when I graduated from my PhD about 4 years ago, I saw an advertisement for CAMDA and I dreamed of participating
Nelly?
someday. Yes, can you hear me right? Okay, so it's truly fascinating to be able to use massive data analysis to extract relevant biology insights. And here we are today representing the winning CAMDA team of the 2023 edition. So it is a pleasure for me to be here with all of you. Let's start with our work. Um, okay, this is a team's work. We were a big team working together for the CAMDA Challenge. And this is our working center here in Mexico. So today I'm gonna talk to you about microbial diversity and metagenomics, the CAMDA challenge, the antimicrobial resistance challenge, the forensic challenge, extra analysis that we did, and also about what did we learn. What is microbial diversity and metagenomics? Well, we know that there are 10 to the 20 stars in the universe. We know that because we work next to the astronomy center. And we also know that there are 10 to the 30 microbes in the earth. But we are just getting to know about that diversity. And for example, before voice works, we didn't know that archaea exist in our planet. It's like living with an alien, and we didn't know that we are living with them. So it is exciting for me and for our team to know that we are learning about the diversity of the microbes that we are sharing the planet to. So, what is metagenomics? Well, metagenomics is the tool that we have to think about why we are learning about this diversity in microbes. Um, uh, for metagenomics, we first have to take an environmental sample, then extract the DNA. After that, we pass all the DNA of these microbes all together to the sequencing. Um, and then we get short reads with the DNA, the sequence of the DNA of all the microbes all mixed together. And after that, we can go through genome reconstruction or to the metabolic and phylogenetic diversity reconstruction. So this is the process. This is how metagenomics works. And thanks to that, we have getting to know that there are some new microbes. For example, this is how SARS-CoV-2 was first discovered in the first place. Well, what it is the CAMDA challenge? CAMDA challenge is the critical assessment of massive data analysis, and it focuses on one of the grand challenges of our time, the intelligent exploitation of big data in life sciences. Well, this year, CAMDA metagenome data consists of 365 samples from 16 cities. These samples were provided by the MetaSub Consortium. There is one day where all the suburban transportation systems in the world is sampled by volunteers, and we have from this effort 365 samples. You can see here is our samples from all around the world. And we also have what we call a mysterious sample. A mysterious sample is a table that contains information about these three bacteria that are clinically relevant, is Klebsiella pneumoniae, Enterobacter ormachei, and Escherichia coli. For all these three bacteria, we have a table with resistance markers, identifiers, and we only know if the marker is present or it's absent in the genome of that bacteria. These are genomes, they are not metagenomes, and all these bacteria were collected from a hospital in one day. So Now that you know what are the data that we are going to work with, now I'm gonna letting you know what were the challenges. We have two challenges: the antimicrobial resistance challenge and the forensic challenge. What is the antimicrobial challenge? Well, what we try to do with the antimicrobial challenge is if we can use the antimicrobial resistance genomes table to predict the city where the hospital is located, where these samples come from. Um, Here, I am showing you some of the reads that are in the metagenomic sample. Some of these reads belong to Klebsiella, Enterobacter, and Escherichia. And perhaps with these reads, we can get some information that when we compare with the antimicrobial genomes table, we can get to know if they are enough similar and infer where is the city, where does these microbes come from. Well, one clue is that hospital is located in a USA city. And now the forensic challenge. Well, the idea is to classify the metagenomic samples by cities. For example, here we have the Bogota city and the Sao Paulo city. Well, we have 3 samples for each city, but the distribution of the abundance of each of the microbes could be slightly different between city and city. For example, here in Bogota, we can see many pink and brown reeds, but in Sao Paulo, there are more green reeds, more abundant. So maybe this small difference can get it into the result that we can classify the samples according to the city of origin. Well, what we do first? Well, we try supervised algorithms, start knowing the origin cities. For example, we divided the full data set in training set and test set. The training set, it's the majority of the data. It could be 80%, 75%. And with this training set, we extract features. And with that features, we try to predict the origin of a new sample. For example, if I showed you this sample at the right, well, we can only observe mainly pink and brown organisms. So with a model, we can conclude that the most possible results is that this sample come from Bogota. So samples are divided in training and testing sets, and we construct a model to predict a class that in this case is a city. We also try unsupervised algorithms to cluster samples in new spaces. Maybe here we are not giving the information that we have a training set, and that training set contains the information of the cities, but here we are trying to clustering, hoping that the samples are similar enough between cities, and then that information allows the samples to cluster together. Here, the Bogota, for example, and the Sao Paulo in another cluster. Well, to understand urban antimicrobial resistance patterns and metagenomes classification by city are 2023 CAMDA challenge. We have, as I let you know, the samples of the mysterious city with antimicrobial markers, and we have the reads from different cities of the world, and we want to use this information to reconstruct which city is when a new sample is presented. What we do first is the typical pre-processing and cleaning of the data. We end up with taxonomic profiles and antimicrobial resistance profiles. This was done by the undergrad student Anton Pashkov. We started with the raw reads. We used trim galore to cure the quality of the reads. The trimmed reads were then passed to megaheat to get assemblies. And after that, we use Kraken 2 to obtain the taxonomic profiles. And we use RGI and the database card to obtain the antimicrobial resistance profiles. Well, I'm going to start with the antimicrobial resistance challenge. Here is the team that worked in this specific part of the CAMDA challenge. And let's find the antimicrobial resistance from the mysterious sample in the USA metagenomes. First, we use CART database to search antimicrobial resistance from mysterious sample in the metagenomes. CART is the comprehensive antibiotic resistant database. It contains genes and it also contains algorithms to detect single nucleotide variations to detect antimicrobial resistance. However, even when we started with 500 antimicrobial markers, using RGI, we could only identify 180 IDs from the antimicrobial resistance in the mysterious sample. Therefore, we created our own database to search the other markers that we couldn't find using RGI. So we built a manually curated antimicrobial resistance database to complement CART metagenomes profile. As I said, we started with 505 markers from mysterious city samples. 180 markers were identified with CART and RGI, but the other ones, we couldn't find them in CART, so we manually curated the database. And with this manually curated 325 identifiers, we perform BLAST versus the assemblies from the metagenomes, and we end up also with that abundance, presence, absence table, similar to the mysterious antimicrobial genome table. So we searched on the USA metagenomes the contigs that were related to the isolates from the mysterious sample by utilizing two different approaches. First, we work with the extraction by genera. From all the metagenomes, the reads from the USA metagenomes, we extract Escherichia, Klebsiella, and Enterobacter, and then we try to obtain the CART and the manually curated identifiers. And the other pathway, trying to get more information, was first we co-assembled all the samples by city, and then we extract by genera. So this is our two different treatments, but for the reads. With these ideas, well, we groped for CART-resistant mechanisms and what we obtain is that CART-resistant mechanisms of Enterobacter and Klebsiella suggest that New York City as the origin of the mystery sample. We knew that the metagenomes had different depths, so that could be a problem. So we needed to normalize in some way. And the first idea was to group by resistance mechanisms, not by all the markers, but only for these 6 main resistance mechanisms in CART, that is antibiotic efflux, antibiotic inactivation, and reduced permeability to antibiotic, all these. And as I said, here you are looking for the Enterobacter, Klebsiella, and E. coli heat map. And this in the horizontal axis is the mystery sample. And here, for example, in Enterobacter, you can see that New York City is sorted by higher similarity, and well, it's the most similar to the mystery sample. For example, in Minneapolis, we can see that there is a lot of presence of antibiotic efflux mechanism, but it's not the case in New York and is most similar to the mystery sample. Also, that same happens with Klebsiella pneumoniae, but that didn't happen with E. coli. In E. coli, the mysterious sample was closest to Denver, and so we didn't know what was happening. So we have the idea that New York City was the origin of the mystery sample, but maybe something was happening with E. coli. So what could be happening with E. coli? Okay. Escherichia is a rare O2 in USA relatively to Klebsiella and Enterobacter. Here we have this 3-axis graph. And as we can see, if we look here for Escherichia, well, there is a lot of Escherichia in these cities. But Enterobacter and Klebsiella, well, they are more distributed and more represented. So maybe what was happening is that Escherichia was less abundant, and the abundance of Escherichia was not enough for us to get a profile for where these samples come from. Okay, then we decided to, instead of using only the mechanisms of resistance, perhaps taking all the markers, the 505 card and manually markers, maybe with that, well, Escherichia mysterious profile is closest to New York. And yes, that's true. Here you can see in the first row the species, Enterobacter, Escherichia, and Klebsiella. Here is Escherichia, and in the second row you can see the cities. The mysterious sample is in pale green. So this is the mysterious samples of Escherichia, and the closest city of Escherichia is New York. So we thought, oh, we get it. New York is the real city from where all the samples come from. Nevertheless, we also noticed that perhaps it's closest because this is the number of positive markers. Well, all those markers that are marked with a 1, that they are actually present in that samples. And we have 180 markers for Escherichia in the mysterious sample, and kind of the same for Escherichia in New York. But all the other samples for Escherichia were really have a low abundance of positive markers. So maybe, again, we are going to have the problem that other cities has less depth in the metagenomes than New York. And well, anyway, New York is closest to Escherichia with the non-supervised method UMAP. We decided to see this in 2 dimensions. Then again, the pale green is the mysterious city. Here you can see the mysterious city. In red is New York City. And well, with the card markers, the 150, the New York City markers are closest to the mysterious, and also with the manually curated markers, New York City is the samples that are closer to the mysterious city. All the other cities are in other places. Also, New York City is closest to Klebsiella with the non-supervised method UMAP. Again, the colors are the same, so you can see New York and you can see the mysterious samples and all the other cities in another cluster. So our analysis indicated that New York City is closest to the mysterious sample city. We were very happy, but then a Pavel email came to our to our email box, and we get to know that the mysterious samples were from Johns Hopkins Baltimore Hospital. So it was not New York, and our fears that we could be in trouble because of the difference of the depth of the samples were true. So we did not get the real answer. It was the Johns Hopkins Hospital from Baltimore. As I said, this could be because New York City has more depth in samples. Here you can see than the other US cities. And because of that, more markers could be detected. Here there is a comparative table from Klebsiella, Enterobacter, and Escherichia. And from 145 markers positives in New York, 41 are in Klebsiella, and all these 41 are contained in Baltimore. All the positive markers in Baltimore are positive in New York. So this makes these cities undistinguishable. We cannot distinguish these cities using these markers, because every time that a marker is in Baltimore, it is also in New York. The same is true for Enterobacter and for Escherichia, there is only one marker in Baltimore that it is not present in New York samples. The same happened with the other treatment, not just directly from the reads, but also from the co-assembled samples. Even here we can get more markers for New York and for Baltimore, but the tendency that the Baltimore markers are contained in New York city markers, it's the same. So these cities, we are not able to distinguish these cities using the tools that we did. Nevertheless, we are working in getting a better prediction of the markers. This is what we think that we can improve, that maybe we are saying, yes, this ROM-A is, for example, present in New York City, but maybe we need to be more strict in the blast parameters or in the RGI parameters, and maybe restricting the positive markers would help us to get better classifications. We have tried several set of parameters, and we have not get Baltimore as the city of origin yet, but I think we are getting improvements in that in that problem. Okay, once that I have finished my talk about the antimicrobial resistance challenge, I'm going to talk to you about the forensic challenge. Well, for the forensic challenge, the goal is understand urban antimicrobial resistance patterns and metagenomes by city. Okay, well, here is the resistance table, and this is what we have been talking about. But now we are going to talk about the profiles to get the origin of the city. Not using now the antimicrobial resistance information, but instead of that, we are going to use the abundance of the taxonomic information. Well, we use two strategies. First, we construct metabolic profiles, and also we construct diversity profiles. The idea is the same. If in diversity profiles, here we have an example of Baltimore and Lisbon, and if I ask you which city is this sample come from, well, maybe the answer is going to be Lisboa, because we do not have green in these samples. And it's the same idea. We are going to construct metabolic profiles, and we are going to try to guess the city using this information. Well, classification is a case of statistical predictions where you start with some variables, for example, abundance, resistance markers, functional information, and a categorical response variable. In this case is the city of origin. We also try other variables like climate information, humidity, something like that. And with this, these are the data, and we try to make some inference to fit some model. For example, logistic regression, random forest, support vector machine, or neural networks. And with this idea, we have a prediction that it is the Y variable. And our goal is to predict the city using a model that takes into account various factors, and then assess the accuracy of the predictions. Well, how can we assess the accuracy? Well, we have several metrics to compare the proposed models. For example, the precision is the rate of right proposed discoveries. Recall is our capacity to detect effects, and F1 score combines it's the harmonic mean of the precision and recalls. So accuracy is our general hit rate. These terms are these measures are in terms of the true positives, what we predict, and it was really from that city. Negatives, true negatives, and false negatives. So remember that we are going to have a training set, and we know the information which samples come from, and we are trying to compare how many true positives, true negatives, false positives, and false negatives do we have. So that's going to be the idea, and each one of the metrics focuses on different types of errors. Well, classifying cities using functional data. This is the team that was working on functional data, was led by Mirna Vázquez Rosaslanda, and we have undergrad students and master students working together for this challenge. Well, we started with assemblies. After we get the reads assembled, we use MiFaser and MetaPsych to get functional profiles. This is an example of functional profile with KEGG. We have the cities, we have the genes, and we have how many reads or assemblies are recognized as to be part of those genes or functional annotations. Okay, in the case of MiFaser, we have tables with easy numbers, and we have a hierarchy, and we use those tables. Well, the idea was to stratify the data, splitting in 85% of training data and 50% of evaluation, 15% of evaluation data. We use MiFaser and MetaPsych functional tables, as I said, and then we stratified in 5 folds, and we run the models random forest, support vector machine, KNN, and MLP. Okay, then we adjust parameters to get the best of random forest and the other machine learning models. We use also a voting ensemble, and with that results, we evaluate the models on the evaluation data. After that, we measure accuracy, balanced accuracy, F1 score, and confusion matrix using the confusion matrix. Okay, the results of this effort is that the MLP classifier was the best one. It outgrows, for example, random forest or SBC. In accuracy, balanced accuracy, and F1. Here is our confusion matrix. And these results are by employing the level 4 functional notation from MiFaser. And this is the best that we get in the functional classification. Everything that you see in the diagonal is right classified, because here in the vertical axis, we have the true labels of the city, and in the horizontal axis is the predicted labels. So for example, here in Auckland, we predict that the 2 samples from Auckland were, and they are actually from Auckland, right? But for example, here in New York City, well, we predicted that 2 samples were from New York City, and no, in fact, they were from Zurich and from Vienna. So what it is outside of the diagonal, it's what we get wrong, and the diagonal is the accurate labels. So with the metabolic profile, we got 80%, about 80% of accuracy. So maybe with the taxonomic profiles, we get something better. So let's talk now about the diversity profiles. This is the team that was working in diversity, and we start first for selecting variables to classify cities using diversity data. Well, I have to say that we, from the Kraken outputs, we got like 20,000 different organisms. So that's a lot to work. The idea is the same that in the functional classifications. We start by, but here the pre-processing was a little different. We first separate O2s by kingdom. Maybe virus has different information than bacteria. We wanted to know and to get those biological insights. After that, we separated by archaebacteria, eukarya, and viruses. And then we also took tables by phylum, class, order, family, and genus. And after that, we tried variable selection using several models. We were following a book that we found about statistics for R in microbiomes. And they use negative binomial regression, and we get inspired by that, and we try to select some O2, some microorganisms that will help us to get the same prediction, but with much less variables. After this pre-processing, we get into the final classification models. We include the selected variables in the model. Again, we use a random forest, a logistic regression, and we calculate also precision, recal, and F1 metrics. We select the best model and calculate the confusion matrix. And also we look if some variable has some biological sense. Okay, what is the idea to select the variables? Well, you could select the variables by the most abundant O2s, by maybe the most the underrepresented ones. I don't know, you have a lot of ideas about how can you select some variable from 20,000 at the beginning. So we decided to use the negative binomial regression to identify differential O2s between cities. So here you have a sample from CT1, from the abundance of some O2, and here is the CT1 mean. So this O2 has some abundance for this city, for the orange city. Now you have the blue city, the CT2, this is the in the horizontal axis, the abundance of the same O2, and you have here the mean abundance of that O2 in the blue city. Here is the orange city mean, here is the blue city mean abundance, and you have the global mean from all these microorganisms, this O2, in both cities. So the binomial regression tries to identify those organisms that their means separate most from the global mean. So in some sense, that would indicate that this deviation from the global mean is an organism, an O2, that separate these two cities. So we search for that in all the cities and in all the O2s, and from 20,000 O2s, we identified 300 that are differential for many pair of cities. Maybe some of them are differential between San Francisco and New York, but they are not differential for Switzerland and some city, I don't know, Bogota or Sao Paulo. Well, we choose those that were differential for many pairs of cities. For example, here we have Staphylococcus. It was differential in 75 comparisons. Bradyrhizobium, it was differential. Well, it was in the top of differential organisms for 46 comparisons, two pair comparisons. So with these 300 organisms, we observed that the selected O2s kept the observed richness structure with less dispersion. Here are the original variables, and here you can see the structure of the selected variables. Well, with these variables, with these 300 O2s selected, we passed this information to the team that in charge of classifying cities using diversity data. This team was led by Dr. Eugenio Balanzario and Daniel Santana, and these were master students of mathematics. Okay, it's in a similar way than the functional classification. We have the abundance table as a starting data, as inputs, and then we proceed with a 5-fold splitting, and then we use 4 folds for training set and 1 fold as test set. Again, we use support vector machine, multi-layer perceptron, random forest. We get the model prediction that we test in the test set, and we calculated the F1 score and the other evaluation metrics. An additional workflow was developed to prevent overfitting and validate the final model. So support vector machine, it was the best model and achieved a macro F1 score of 0.96. 96% is much better than the 80% that we get before with functional classification. So here you can see the support vector classifier is the best method. It's best than K nearest neighbors and the multi-layer perceptron. And this using only these 300 variables. At the end, we use all the kingdoms together. We didn't get better results restricting ourselves to bacteria, virus, or eukarya. It was best to use all the OTUs, but only the 300 selected variables. Here you can see again our confusion matrices. And as you can see, there are a few values outside of the diagonal, and this is good because a perfect classifier, you will always suspect that there is something wrong or you overfit the models. Okay, after we finish with the challenge proposed by the CAMDA organization, well, we also were very interested in some extra analysis and to get more information, because at the end, what this challenge is about is that you can learn, and your team and your students, to get clean big data analysis, well documented, and to explore what information you can get about these biological data that are provided to you. So what we did is topology, regression, and metapangenome to enrich the conventional microbiome analysis. This was the poster CO6 in the CAMDA conference, and this work was done by Aide Perugero and Shadai Guerrero. They both are postdoctoral researchers in mathematics. Okay, what is topology and topological data analysis? Well, topology helps us to study characteristics of geometric structures. For example, here you can see a ball that has no holes, but a cup and a donut, they all have at least one hole. And if you perform a continuous deformation, you will get one hole there. So topological data analysis is a technique that uses concepts from topology to analyze complex data and find patterns and structures that are not apparent at first glance. TDA has been used recently in genomic analysis. So can we use TDA to predict something about the mysterious sample? Well, perhaps not the origin, but maybe something about it. How does TDA work? Well, you start with a cloud of dots that you don't know how are they connected, but you move a parameter. This is a radius. It's like you inflate the dots and you see, well, are they closer than this radius? Some of them are, so you start tracing, drawing vertex between them, connecting them. And this is called a connected components, and you are here getting the information of how many connected components you have. This is the H0 is how many connected components, and H1 is how many holes do you have. Here you do not have any hole at the first figure, but once you are inflating the radius of the distance, you can see some holes are occurring between how are you connecting those data, and this hole is maintained a lot of time. So this could mean that, well, perhaps the form of your data is similar to a donut that has only one hole. So TDA is like keeping information about how many holes are you getting when you are connecting your dots in using some distance. So for example, here you have 4 nodes, 4 connected components at time 1 in the case of a tree. This is why we see 4 red bars and no holes, because they are no holes in the connections. Trees do not contain holes, so they can be described as several connected components. Here you have, this can be some organisms, for example, and the first distance threshold is connect 2 of them. So now you only have 3 connected components, 3 bars. One of them are dead at time 2. You only get 3 connected components. Well, if you relax the parameters, then you have 3 dots connected in a tree, and now you only have 2 connected components. That's why in time 3 you only have 2 connected components, and the other ones died sometime before. And at the end, you have a connected tree with no holes between your connections. The 4 nodes get connected with a relaxing distance. In time 4, you have a tree, and this the old friend, the phylogenetic tree. Nevertheless, when there is horizontal gene transfer, some genes' history is not tree-like, and this can be captured with holes. Here, perhaps these genes are are closer together than the species tree would let you suspect. So now you get a hole between these nodes, and this hole you can see it here, for example. So with TDA, we can get an idea about histories that are not tree-like. So TDA approach with a threshold of 0.2. New York City is joined to mysterious samples. So here you have, at the beginning, no samples are, well, some samples are connected, but for example, here are connections between the mysterious samples, some connection between New York City, but that are dispersed in a cloud. But with threshold 0.2, well, now they are connected. And you can see some holes here. Well, there is some horizontal gene transfer between Klebsiella, Escherichia, and Enterobacter, and we think that may be relevant if you are looking for nosocomial infections. These are the data of all of them. Here we have the connected components, and in blue and green you have the holes. And when separating these diagrams by organism, we know that Escherichia has some holes. So there could be some markers that are transferred horizontally, but Enterobacter does not show horizontal gene transfer against other Enterobacters. Okay, this is what we do about topological data analysis. And maybe we have the next analysis is we have other types of data to classify cities. Perhaps the taxonomical distribution or the functional distribution, it's more related with bioclimatic variables or with demographic variables. So with this idea, this team, Andres, Camila, Miguel, and Aide, try a Dirichlet regression to relate abundance of microorganisms to city covariates. And it seems to find some relationship, especially with climate variables. And the last analysis that I'm going to present to you is the metapangenome. Metapangenome, it was proposed by the ANBIO team, by Meren researcher team. So metapangenome does a comparison between reads against pangenomes to show differential read abundance. It's like a pangenome that compares the abundance of some gene families between a set of phylogenetically related genomes. But now what you are trying to do is to get this extra layer of information that you obtain from metagenomes. So it's like you can have several strains. You construct first a pangenome, and then you compare the reads against the pangenome that you have constructed before. So metapangenomics is the combination of genomic data and environmental samples. So metapangenomes compares reads against pangenomes, showing differential read abundances. So first we get some genomes, all the genomes that we could find from Baltimore, Nueva York, San Francisco, all the cities that we got from the CAMDA challenge. And we compare this pangenome against the reads. So as I told you, first is the pangenome construction, then we use reads alignment, and we get some classification of the reads as core or accessory if they are present in the different metagenomes. Here, the pangenome revealed that Klebsiella genomes are divided in two groups. I feel that's because of their, well, the resistance could be mechanisms. But anyway, pangenome splits Klebsiella in two groups from different cities, Baltimore, Nueva York, and San Francisco. After that, Baltimore metagenome reads has some differential gene families. We still have not get deep in these families, but this is something that we are planning to do. But here in Baltimore, there are families that are present in Baltimore and Minneapolis, but not in San Francisco. New York City, also because of the depth of the metagenomes, has a presence of reads in almost all families of the pangenome. But we are planning to explore this differential. These are the reads aligned to the pangenome, and we would like to explore well, differential reads when compared to the pangenome. So what did we learn? In conclusion, we learned that to predict the mysterious city, we use card profiles, which determine the closest city as New York, even though the true city is Baltimore. This discrepancy could be because New York City has more depth in 2016 samples. And we developed a pipeline that allows us to select a set of O2s based on abundance data, enabling us to effectively address classification problems with less variables, while also providing some biological interpretability. For forensic challenge, we kept less than 300 variables and applied support vector machine to classify, and it got 96% on F1. And our results are reproducible, or we are trying our best to get to that point. We have a GitHub repo. We are making a CAMDA and Docker containers to share all our work with the future participants of the CAMDA challenge. And we made pertinent documentation and CAMDA repositories. You can check our repo with this QR code. And the last thing that I have to do is to thank all our team. As I said, we have mathematicians, biologists, and computer scientists from undergrad level until a team of researchers from our country. We are a Mexican team. This year, 2 Mexican teams participate in the challenge. I feel like this is the first time that Mexico participates, so we are very happy and we thank the organization. And we also have to thank all our sponsors, UNAM, CIMAT, the Center of Mathematical Science, the Institute of Radio Astronomy, Sinvesta Blanquevio, and the Mathematical Mexican Society, because thanks to them, to the funds, and to the servers, because we do not have a big server, we have to ask all these centers to share with us 5 servers to process all these 400 metagenomes data. So we really thank to them. We also thank the GAPA and PAPIT that supports our research with some grants. So thank you very much. And I'm open to questions.
Thank you, Nelly, for a very rich presentation. One thing which maybe it's a nice to tell is that this presentation, your work won the CAMDA trophy, And you say that you are using this MyFaster tool, right? And the MyFaster is from the lab of the Jana Bromberg, and in fact, the Cheng Sheng Chu, when he was presenting results of his work, where in fact they were introducing MyFaster in Chicago at CAMDA, they also won the CAMDA presentation. So your colleague won the cheese head, and he won at that time because it was in Chicago. It was Al Capone head. But going back to the science, there's a set of questions. I will just start from the top. So there is a question. Can you elaborate more on the manually created database? Do you plan to release it publicly?
Uh, which one is that question? I'm sorry, I was seeing the
Uh,
chat, but can you?
I, no, I there's a Q&A. So you were talking that you were using the card database, but also you were using some manually curated one. So the
Yes.
question can you say a bit more about this database and also whether you plan to release it, to make it public?
Oh, yeah, sure. Well, the mysterious sample contains the names of the genes, but CART um, didn't predict these genes because they say the model is not available. I don't know why. I guess I have to research more about antimicrobial resistance. But what we did is that we look for those genes for Escherichia, Klebsiella, and Enterobacter in NCBI. We get all these sequences in a FASTA file. This file is available in our repo. That's the idea of make everything reproducible and shareable. So you can get this file in our repo, of course. I I don't know if it deserves to be published somewhere else, but of course it's available for you to use it there.
Okay. I guess that maybe I don't know what you are planning to submit as a follow-up of the CAMDA, but maybe then you can also attach it so then it can be available, or at least the point to it. Okay, so then the second question this time from Kinga is it possible to apply multi-omics approaches? So just move beyond gene annotations to improve your predictions.
Yes, I guess. Well, the CAMDA challenge, forensic challenge, is to get to know where the cities come from, where the samples come from, and what you get is well the raw reads. But you can use whatever information you can get from those raw reads. It could be taxonomic as we did, it could be functional as we did, but as long as you have some features that you can set in a table, well, you can try the classifiers, and if you get more information science, other omic science, that for sure would help the predictions. You can try it separately, as we treated the functional and taxonomic predictions. But I have seen that in the past, some teams combined these approaches. They combined taxonomic and functional classification, and they got better results. So we will have to try if separated or together, but another layer of information will always get to better results.
Okay. Another question is about the metrics for the distance. I mean, you were showing the UMAP plot when then you were saying about the proximity and based on this the classification. So have you also tried the different better metrics to show distances? As not all the approaches which do the dimensionality reduction, they keep the scale for the distances. So have you tried something
Um,
else than UMAP?
um, no, we didn't try something else. In UMAP, well, and directly in Python, we calculated only the Hamming distance and several cluster analysis, but we are on time. So if you let me know your suggestions, we can still try.
Okay. Um, Yeah, so I think then at some point we can also have a chat. So maybe you can think about something else to try. Another question, because you were saying that the fact that the New York, in fact, is winning in many cases might be because there's more samples from New York. So have you tried rarefactioning to mitigate the depth sequencing problem that you have? I mean, to use the subset of the data and then try everything again?
Yes, we tried rarefaction, but it didn't help to the prediction.
Okay. Okay, and the last questions from the Q&A is what the for the TDA approach, what was the input data that you used to generate the network?
Which network?
I don't
Yeah, I
know.
see. For the TDA approach.
TDA approach, yes.
Mhm. Okay, the input data were the mysterious sample with the matrix of zeros and one. It's kind of a resistome in the antimicrobial resistance, but we also added, we predicted the antimicrobial resistance of all the genomes that we found in NCBI from those dates and from those cities. We use the zeros and ones card antimicrobial, well, all the 505 antimicrobial markers from the mysterious city were searched for the metagenome samples. We also use the mysterious sample and we also use the NCBI genomes. But what I showed you today, it's only the TDA results for the mysterious sample, because we think this has more biological sense because all of them were collected together in one place, and so we can talk about horizontal gene transfer because bacterials are living in that place. And we feel that if we mixed this data with the metagenomes and with the NCBI genomes from other places, maybe the biological meaning would be, well, would have nonsense. So
Mhm.
what you see today is uh, the markers from the resistance markers from the mysterious city in the TDA network.
Okay, just there's one more question, but before I will go to this, just one information that the mysterious samples, I mean the isolates from the hospital, they were collected over a few years. So it was not that they were coming from, I don't know, the same time point. So they might, that's why they might differ
Okay.
in the markers. So if I could remember, and probably will release this information with the next challenge. I think they were collected over 4 years, so they are not from like one time point, but they are so they can be diverse. So there's one, and I think last questions. Were you able to detect all AMR markers present in the isolates in the metagenomic samples?
And no, not all were present in the metagenomic samples. As I said, the 180, it's the markers that are present in New York City, and New York City is the sample that has more positive
Mhm.
markers. And well, other cities has other markers, but for example, Baltimore, all their markers are contained in New York City. So not all the 505 were detected in the samples.
Okay.